About the transduction. Today, we are going to discuss about the transduction process in bacteria. This is again another third technique where the DNA is getting transferred from donor bacteria to the recipient bacteria through a bacteria called as viruses. So we are having certain viruses which only infect bacteria. These bacteria are called as bacteriophage or phage. These bacteriophage insert its DNA into the cell, into the bacterial cell, from where they are able to transfer its bacteria's genome or bacteria's DNA into the new bacteria. So there is interaction of bacteria and the viruses. If we see the literature meaning of this transduction, trans means across, duction means to pull. So naked DNA is transferred through the process called transduction via the bacteriophage. This phenomena was first discovered in Salmonella species in 1952, and two scientists observed this phenomena, named as J. Ledenberg and N. Zinder. Viruses have their coat that is called as capsid, and in this capsid we are having core of nucleic acid. We are having, when these viruses attack bacteria, they insert their nucleic acid into the host where they reproduce themselves, that is within the bacteria cell. To infect with this bacteria, these phages should have ability or capability to get attached to the receptors which are present on the cell wall of bacteria. Then when they secrete the enzyme which creates a hole in the cell wall of the bacteria through which the nucleic acid of the bacteria enters into the through which the nucleic acid of the viruses enter into the bacterial cell. So phage has having some enzyme which weakens the bacterial cell wall. The protein coat of these viruses remain attached onto the walls of the cell wall. When the nucleic acid enters into the cell, it follows two different pathways. This phage, when the virus is inside when the viral genome is inside the bacterial cell, they may be virulent. We call this as virulent phage, or this phage will remain silent for some time. So we call them as a temperate phage. That is virulent phage and temperate phage. Virulent phage is having ability to cause infection and eventually destruct the bacterial cell. When the phage nucleic acid enters the cell, they interact with the genome of the bacteria where they direct synthesize their own proteins, own nucleic acid, and own capsid proteins, which will be used for its own formation. So they disrupt the host cell's DNA. They only produce their own nucleic acid and proteins. So these proteins, nucleic acid and capsid proteins come together and they again form a complete phage in the bacterial cell. When the bacterial cell becomes filled with 100 or more phages, phage again secrete the enzyme which rupture the bacterial cell wall and releases these phage into the environment, from where these phages again infect another bacteria and again cause the disruption. So this cycle is called as lysis, or we call this as lytic cycle. You can see in the diagram on the extreme left-hand side, where the phage is getting attached onto the cell wall. Now the second step is this phage is outside only, but they are able to insert a genetic material into the host bacterial genome. Then there is the formation of all the necessary items, which is utilized to form phage again. Now another diagram is showing, another picture is showing the phage is ready to get released from the bacterial cell. Now in the next step, the phage is getting released. Now this phage is ready to get attached onto the new host. Now the second one is the temperate phage. This does not this cause occasionally disruption or infection to the bacterial cell. What these phages do, these viruses insert their DNA into the host genome, then they incorporate into the bacterial DNA, where it gets replicated. These phages produce some repressor, some enzymes which does not lead to the disruption of bacterial DNA. It means जब ये इंसर्ट हो गया वायरस का जीनोम किसमें बैक्टीरियल सेल में, तब वो कुछ रिप्रेसर सब्सटेंस, कुछ ऐसा सब्सटेंस प्रोड्यूस करता है जिसकी वजह से डिस्ट्रक्शन ऑफ बैक्टीरियल डीएनए नहीं होता है, एंड phage DNA remains within there, so वो अपने पार्टिकल्स वहां पे नहीं बनाता है. That is, phage DNA does not direct the synthesis of phage particles. जो phage को बनने के लिए पार्टिकल चाहिए रहते वो वहां पे नहीं बनाते. They remain within the bacterial genome, and this condition is called as prophage. When the phage DNA, virus DNA, is silently within the genome of the bacteria, this is called as prophage. It means when this prophage is within the bacterial genome, they don't create the lysis of the cell. That is called as lysogeny. And the cell which contains this lysogeny DNA of the bacteria is called as lysogenic. Is said to be, the cells containing the prophage are said to be lysogenic. Now, there are several different environmental conditions or several ways by which we are allowing this lysogeny or lysogenic cycle to convert it into the lytic cycle. Actual reasons are not known, but there must be some reasons which is behind why the lysogeny is converted to the lytic cycle. These temperate phage can replicate with the bacterial chromosome, or they are having ability to replicate independently, so that they will be assembling to form a new phages. When the condition arises where the lysogenic cycle is getting converted to lytic cycle, at that moment there is the packaging of DNA, virus DNA into the capsid, but sometimes it is possible that they might take DNA from the bacteria into the capsid. So there are two processes which describe this transduction. First is the generalized transduction and another is the specialized transduction. As you can see in the diagram, when the phage enters its genome into the bacterial cell, then the two cycles can occur, lytic or lysogeny cycle. When this phage DNA enters, remain silently within in the host cell, that time this is called as lysogenic cycle. Or when this genome creates disassembly of DNA of bacterial cell and form their own proteins and DNA, this is called as lytic cell, as you can see in the diagram. Now the specialized transduction. Several lysogenic phages are known to carry out this specialized transduction. ये transduction है, specialized transduction, it is carried by many lysogenic phages, and the best study till now is lambda phage, which interacts with the E. coli cell. So what they do, these lambda phage, they usually insert at a specific location. They specifically insert within in the chromosome at a specific location. Mostly it is seen that when these phage, like a lysogenic phage, insert into the bacterial genome, it has been seen they have inserted their genetic material either at GAL gene. This GAL gene controls the lactose use, or the biogene. This biogene controls the biotin synthesis in bacteria. The GAL gene the GAL gene and biogene are part of operon system of bacteria. When the bacterial cell contain lambda phage, so this lambda phage induces entry of lysogeny into the lytic cycle, and the genes of the phage from the loop are excised from the bacterial genome. Then the lambda phage, ये lambda phage है किसमें बैक्टीरिया जीनोम में, ये डायरेक्ट करता है कि हमारे जो phage particles है, like their capsid proteins, their neck, they allow the formation of their own machinery, then the cell get lysed. This is the specialized transduction. It has been seen that sometimes the GAL gene might be incorporated into the phage particle. As I have said earlier, sometimes these phages are having ability to take some part or take some DNA genetic material from the bacterial cell. They get incorporated into the capsid. So it has been seen that the GAL gene might be incorporated into the phage particle. But when this phage infects another bacterial cell, they transfer this GAL gene. So this bacterial, the neobacterial infector, will be having two GAL genes, and this is called a specialized transduction. You can see in the diagram, the first DNA integrates into the chromosome. Then the next step, prophage deintegrates and pick up the piece of bacterial chromosome. Then replication of viral DNA and the destruction of bacterial DNA takes place in the cell. Then the virus capsid synthesis takes place. After synthesis, the assembly of this capsid takes place. Then the lysis of bacterial cell occurs, where these phage cells are getting released, and now they are within the environment where they can infect the new bacterial cell. Now the next step is crossing over, and stable genes transfer into the bacteria recipient cell chromosome, as we discussed about the GAL gene right now. Now the generalized transduction. When the bacterial cell are having phage DNA, they enter into a lytic cycle. This lytic cycle is occurring because of the phage enzymes, which breaks host cell DNA into many segments. As the phage directed the synthesis and assembly of new phage particles, it packages DNA by the headful. That is, more amount of DNA is present in the head of the viruses. This allows a bacterial DNA fragment occasionally to incorporate into the phage particle. Likewise, we have seen the DNA from plasmid or other viruses which get packaged into the phage head. Now this phage particle will infect another bacteria and leaves the infected host. it may infect another bacterium and transfer the bacterial gene through the generalized process. And this technique is called as generalized transduction, as you can see in the diagram. Now, if we talk about what is the significance of this transduction, first, the genetic material from one bacterium to another is getting adapted. If there is transfer of genetic material from one bacteria to another bacteria, that leads to alteration of the genetic makeup of the another bacterium. In case of specialized transduction, the gal gene, if the cell is not having ability to have active gal gene, or if the bacterium is not having the gal gene in it, due to this transduction process, they will be acquiring gal genes, then they will be able to metabolize galactose, which will be present in the media. So means एक जीन अगर बैक्टीरिया में गैल जीन नहीं है, तो due to this specialized transduction हमें उसको acquire हुआ गैल जीन. Now this bacterium is also having ability to metabolize the galactose. This transduction demonstrates a close evolutionary relationship. This is the mechanism where we are relating the evolutionary relationship between the prophage and the bacteria host. It is only because they must be having quite similarity of sequence, then only there will be otherwise, if there is no sequence similarity, then prophage would not bind to the bacterium chromosome. होमोलॉजी होती है ना, जब होमोलॉजी होगी तभी क्रॉसिंग ओवर होगा, तभी एक दूसरे के साथ इंटरेक्ट करेंगे. अगर वहां पे वायरस जीनोम और बैक्टीरियल जीनोम की होमोलॉजी नहीं होती तो इंटरेक्ट नहीं कर पाते. It means they are having homology, so this understands our evolutionary relationship. This completes your transduction process.
So in
Today,
next
we are going
lecture,
to discuss
we will discuss about the biofilms. What are biofilms, how they are formed, and what is the significance of biofilms in day-to-day life. Thank you.